series hosted by IMB, and okay. So there are other 26 other centers that are that love RNA as much as we do. So if you're one of them that are not here, actually, you can also talk to us and see if we can have great talks from your side as well. Okay. Just as a reminder, that all the talks are recorded, so you don't need to record them yourself or take pictures of the talks. You can always check them up on our YouTube channel. And yeah, we already had a bunch of other seminars since 2020, and we'll continue to do so. And you can check them all on the YouTube webpage. So today we'll have two talks. We will start with Miona, and she's a PhD student now at the Julian Konig's lab here at IMB in Mainz, and she actually did her bachelor's and master's in the University of Belgrade in molecular biology, and now she will cue us in a bit about M6A. And yeah, Miona, when you're ready, you can start.
Thank you, Joe, for the introduction. As Joe said, at the moment I'm a PhD student in Julian Konig's lab. And in this talk, I will tell you a little bit about M6A modification and what it does when it's located in the coding region of the transcript. So in this talk, I really don't have to introduce this molecule. So this is the molecule of the RNA. And what we are interested in Julian's lab is how it is impacting the gene expression. And this is how maybe usually the molecule is depicted, but in Julian's lab, we see it more like this, covered with mRNA modifications. And there is more than 170 modifications in RNA, and they are changing the lives of RNA in basically every level and every step. But if I'm being completely honest, this is how we see the molecule. So we are really concentrating on the M6A modification. And what is M6A? It is the methylation of the adenosine in the N6 position. And it is predominant internal mRNA modification. It is written in the nucleus by the core protein complex metal 3 model 14 in a sequence-dependent manner, where the A that is methylated is always followed by the C. And it can be read either in the nucleus, where the predominant reader family are YTHDC proteins, and then it can have impact on RNA export or splicing. And it can also be read in the cytoplasm, where predominant reader family are YTHDF readers. And once they are reading the M6A, they are impacting the stability. And the RNA stability is exactly what we were interested in this project. But before I start now talking about stability aspect, I would like to introduce a couple of more things. So in order to be able to discuss any influence of the modification, first we needed to know exactly where this modification is located on the RNA. And in the last 15 years, there was effort from a lot of people and a lot of groups for mapping the M6As in the more or less specificity. And there is this meta profile that always comes up where we have the enrichment of the M6A around the stop codon, while it's a bit restricted in the CDS. And that was a big question, why or how we see how this happens that we have it less in the CDS. And this was answered by these 4 great papers, the beginning of now already last year, where they discovered that on the exon-exon boundaries, there was exon junction complex sitting, and it was preventing the M6A writer complex from depositing M6A, which made it basically very difficult for deposition in the short exons. And that did answer the question why we see this restriction in the coding region, but it still left the question why, what is the physiological role of this? And also then what we were interested in, does it mean that maybe M6A can have different roles if it's located in the coding region or in the untranslated regions? And in order to start answering our questions, we first looked into the publicly available data set of the mRNA half-lives in HEK293 cells, where we do most of our experiments. And so in this plot now, you're looking into changes in the mRNA half-lives, and if the box is going more up, that means that half-lives are longer, and more down, that half-lives are shorter. And then we divided the transcripts by them having the M6A sites in either 5' untranslated region, coding region, or 3' untranslated region. And then inside of all of these three regions, we further stratify them by the number of M6A sites that you can see by the color shading. So the more darker color means that they have more M6A sites in this region. And there we observed that, as expected, the more M6A sites in all of these three regions transcript have, the shorter half-lives they have. But here we also observed that this is really the most striking and the most strong in the coding region, which already a little bit made us look more closely in the coding region. In order to now look at this more dynamically, we made use of the metal-3 inhibitor called STORM2457. So what it does, it inhibits metal-3, one of the writer proteins, and it inhibits the deposition of the M6A. And we did it in a dynamic way. So we did a time course experiment and performed RNA sequencing. And now in this plot, what I'm showing you is not changes in half-lives, but changes in transcript abundance, but everything else stays the same. So if upon M6A inhibition, upon STORM treatment, we had more of the transcript was stabilized, we would have them going up. And here now we were really, it was really interesting to us that after 6 hours of M6A deposition, we could see the stabilization with increasing number of M6A sites only in the coding region. While we couldn't see any change, it doesn't matter how many M6A sites they had in 5' or 3' UTR. And upon longer treatments, upon the depletion for 9 and 24 hours, now already also transcripts in 5' or 3' UTR were starting to have the effect. So we observed that CDS M6A sites trigger rapid and efficient mRNA decay compared to other transcript regions, and we wanted to investigate this. So since we are talking about something that is really specific for the coding region, first thing that is it involved translation, something very specific for the coding region. So we made use of a translation inhibitor called emetin, and we inhibited translation. And now we are looking into log twofold change in transcript abundance upon treatment with emetin, again, RNA sequencing data. And we could see this specific effect of CDS M6A sites. And even more importantly, we performed translational inhibition also in M6A depleted background, and here this effect was gone, which now really made us think that maybe translation can be coupled with this CDS M6A specific decay. And in order to try to investigate it further, we made a reporter assay, where we have our reporter has the GFP coupled to the P2A site with the insert of our choice, which was the last part of the CGRRF gene, where it was one of the genes that was upregulated upon our 6-hour STORM treatments, and it has this nice 5 M6A sites in the last exon. And how we designed the experiment is that we transpected in our HEK cells in presence or without presence of our M6A inhibitor, and then performed flow cytometry to measure levels of changes of GFP. And now in the plot, I'm showing you log2-fold changes in GFP signal upon M6A depletion. And we were really pleased to see that when we transfect our cells with this plasmid and remove M6A, we can see strong stabilization. And in order to be sure that this stabilization that we are observing really comes from these 5 M6A sites, we also made a mutant where we mutated all 5 of them. And here you can see that the effect of M6A removal is lost. And even more importantly, we made mutants where we moved the stop codon to now be upstream of the M6A sites, moving them from the coding region to the 3' UTR. And as you can see, still the effect of M6A deposition were not existing, which now really proves that translation is coupled and that that this pathway of decay is going through translation. And in order to now try to answer more mechanistically, how can it be, we performed ribosome profiling. And just in a couple of seconds, how we do ribosome profiling, what it is, so we have our RNA that have ribosomes on it, and we degrade the RNA, and then only the RNA that is now protected by the ribosome will not be degraded. Then we also remove the ribosome, and then we have this RNA footprints, ribosome footprints that we can then sequence, and then we can know exactly where the ribosomes were sitting on the mRNA. And if we have more reads from one location, that means that the ribosome was there longer, there was more ribosome sitting there, indicating the pausing or stopping of the ribosome. And then we looked into the normalized 5' end signals, and we put it in the position relative to our M6A sites. And we were really pleased to see here, you can see the peak on the position around -16, which basically is the position where of the 5' end of the footprint, if the M6As would be located in the A site of the ribosome. And we were even more pleased to see that once we depleted our cells of M6A, the signal was lost, which was then really showing that this pausing of the ribosome is caused by this M6A site. And this leads me to my first summary slide, that if we have the mRNA that has M6A sites in the CDS, and this mRNA is being translated, once M6A site is in the A site of the ribosome, it will lead to ribosome pausing, and it will influence RNA decay. But still, what is the mechanism of the decay itself? So in order to answer now these questions, I will go back to the introduction, and you can remember that I mentioned you that one of the main breeder of families are YTHDF proteins, and especially YTHDF2 protein. And we were interested, is YTHDF2 still now needed or not in this pathway coupled with translation? So we first performed YTHDF2 iClip experiment so that we can know exactly where YTHDF2 is binding to the mRNA. And we could see this specific Drach motif, which I told you in the beginning is the motif where M6A modification is put. And then we used this binding map of YTHDF2, and we made a very similar plot to the one I showed you in one of the first slides, where we are looking into log2-fold change in transcript abundance upon M6A depletion, upon 6 hours of M6A depletion. But now we separated transcripts, and here we are looking only in the transcripts where YTHDF2 is binding them in the CDS. And as you can see, we still have this strong impact of the CDS M6A sites. But then if you are looking to the transcripts that don't have YTHDF2 binding in the CDS, this effect was lost, which then, along some other experiments that I will not be showing today, really led us to believe that YTHDF2 is somehow involved still in this process. And in order to answer where, we first went back to the ribosome profiling, because first idea was that maybe it would lead to ribosome pausing somehow. But then when we overlapped the footprints with or without YTHDF2 binding, there was really no change there. Which then led us to believe that it comes more downstream later. And to try to understand where and how, we then tried to look into the, we looked into the proteins that it is interacting. So we performed the proteome proximity experiment of the YTHDF2. And together with ribonucleoprotein granule interactor, So now here I'm showing you geotherms for YTHDF2 interactors, and very highly up we could see this P-body proteins. So P-bodies are cytoplasmic granules that are formed by phase separation, and where translationally repressed transcripts are located together with decay machinery. And then also in already published data, we could published papers, there is a lot of connection between M6A sites in P-bodies, which then led us to maybe think that this transcript somehow could be, that YTHDF2 could contribute in the context of P-bodies. And in order to check that, we made use of the publicly available data set of transcriptome-wide transcript localization. And we tried to see if, to look into the transcripts that are enriched in the P-bodies. So here on the plot, we are looking more to the right, transcripts that are more enriched in the P-bodies, and more on the left that are not. And we made use of all the RNA-seqs that I was mentioning to you before, and we defined target sets. So we looked into the transcripts that are upregulated in all of these treatments. So upon M6A depletion, 6, 9, 24 hours, translational inhibition, YTHDF2 knockdown, and if they were upregulated, we defined them as the targets of these proteins. And as you can see here in red and orange, the targets of the CDS from the M6A depletion for 6 hours and from Ametin were strongly enriched in the P-bodies. And in order to look at this in more detail, we also performed a microscopy experiment where we did immunofluorescence staining for the P-bodies using DCP1A marker. And we looked at the number and intensity of the P-bodies in the control DMSO and also M6A depleted background. And we could see that when we deplete M6A sites, we have significantly reduced number of P-bodies. I will not show you the plot for intensities, but it looks fairly similar. So we also have significantly reduced intensity of P-body signal. So now we are pretty confident to say that CEMD targets localized to P-bodies, but still we didn't answer the question which other factors are involved in the decay. And once again, we went back to our main reader proteins, and we went back to the already published mechanism in which M6A is leading to decay, where it was published either through interaction with C01, where it leads to deadenylation, with HRSP12, where it leads to endoribonucleolytic cleavage, or through UPF1, when it leads to decapping. And then we tried to find publicly available data sets for RNA-seq where all these proteins were depleted, either by knockdown or knockout in our cell line, and to see if in any of these data sets we would see the CDS-specific effect. And then when we looked into the effect of the C01 knockdown, we could see that the effect exists in all 3 transcript regions. From the HRSP12 depletion, we can see only effect in 3' UTR, and similarly as to C01 with the UPF1 knockdown, we could see effect in all 3 transcript regions. So neither of these candidates seemed specific for our CDS mechanism. But then we still looked, since we are talking about localization in P-bodies, and one of the main proteins, decay proteins in the P-bodies is DCP2. We also checked the publicly available data set from DCP2 knockout. And here we could see this CDS-specific effect of the increased number of M6A sites in the coding region, which then made us believe that it might be linked to the decapping via DCP2. And one more thing that we did in order to try to distinguish our pathway from other known pathways, and also we made use of our already defined set of targets, where we defined 6-hour targets of the M6A depletion of the 6 hours. And we looked how these targets are acting in all of these data sets. And we could see that in DCP2 depletion, after DCP2 depletion, translational inhibition and YTHDF2 knockdown, we can see that these targets are upregulated while they remain unchanged upon CNOT1, UPF1, or IRGR-SP12 depletion, which now really made us confident that our pathway is separated from other known pathways, and it is coupled to translation by THDF2 and maybe DCP2. There we need to do more experiments. And this leads me to the summary of the talk already. So once we have the mRNA molecule that has M6A sites in the CDS and is being translated, once a M6A is in the A site of the ribosome, it will induce ribosome pausing, and these transcripts will be translocated into the P bodies, where they will be decay bound by THDF2 and degraded in the pathway that we named CDS M6A decay, or CMD shortly. And the more factors here would be needed to distinguish in the future work. And something that I was not talking a lot in this presentation, but that can give idea about the physiological relevance of this pathway, are some of the targets that we could see, some of the targets of this pathway. So we could see the M6A target pathway transcripts that are so self-regulating themselves, basically. Also, there was a lot of developmental genes such as SOX2 or KLF4. And as well, one interesting group that we observed were retrogenes that evolved through retrotransposition of their already spliced parental transcripts. And therefore, for the cell, it's really important to keep them silenced, which means that maybe even this pathway can safeguard the transcriptome somehow. And with that, I would like to thank Julian and Kati for this amazing project. It was really a pleasure working on it, and always nice discussion with them. And all people from my lab, but especially Peter, because with him I've done most of the wet lab parts in the project, and from Kati's lab, Joe, because he did all the bioinformatics stuff in the project and really made it possible. And also other collaborators, Mihaela, for giving us the plasmid, which we modified in order to define our reporter, Stepanka, for the experiments with proteome proximity, and show the data today, but they were also involved. As well as my TAC members, of course, and core facilities here at IMB, because they make our life much easier. And also all of you for your attention. So I'm open for questions now.
Thanks a lot, Miona. So there's, let me remind you that if you have a question, you have to write it in the Q&A section. And we already have a question here. So Ildar is saying very interesting work. How do you envision M6A causes translation pausing?
Yeah, this is a very good question. How I think it happens, I think it just with the it slows down the reading of the codon, basically, or this reading of the codon of the ribosome. So I think there was recently a paper in bacteria that really showed that when there is M6A on the A site, somehow it can move the conformation and then make it more difficult for reading. So I think, so we also don't believe that it really stops now and doesn't move further. So I think it just slows down it enough, basically, so with the reading. I hope I managed to answer it.
Okay, so we have another question here. Are RNAs that are bound by YTH-DF1 protected from YTH-DF2 mediated degradation?
Yes, another nice question. So I am not sure. So we never did experiments that would really control for both of these things in the same time. But what I can tell you is that we were always doing more or less in parallel experiments with just YTH-DF2 knockdown and with knocking down of all three proteins. And the results are almost the same. So maybe slightly stronger effect we have when we knock down all three of these proteins. So I would not say that they are protected.
Okay, so another question here. How many transcripts are affected here? I think I am reading as well.
Yeah, now I'm afraid that I will maybe lie, but I think our CMD targets that are defined are, I think, around 150 to 200, but I am not 100% sure now that I said it correctly, so I would need to check. I can get back to you.
Okay, another one. Do you think M6A in 3' UTR have different functions versus M6A in the CDS?
So yeah, I don't think so. So I think so far it seems that it always actually leads to decay. I think it's more than maybe once in the CDS are leading to more fast decay. And I think the transcripts that are affected are different. So once we run the geo terms, we don't get the same the same terms. So with our CMD targets, we can see more important to the moment when cell is changing the state. So I think it's more where you need the rapid effect that will also maybe self-control it. It's more this CDS effect, and then in the more long-term housekeeping genes, it would be more the effect of also 3' UTRs. But I think, yeah, something like that could be interesting, that maybe some of the M6A sites are affecting the other or something like that, but I have nothing that could say anything like that.
Okay, I don't think we have any more questions, and maybe we could stop it here. So thank you, Miona, for the great talk.
Thank you.
And now we go to our other speaker, Miya. Miya, maybe you could. Oh, you're muted. Maybe unmute.
So I may start now.
Yeah, let me just introduce a bit. So I know that you went around the world doing your studies. It's a bit hard, but you did your PhD in Rennes in France, and then you went on to do a postdoc in Switzerland, right? And after that, you landed here at IMB at the Keller-Valsecchi group. And I presume that this was also where you did most of the work that then culminated in the talk you're going to do here now. So let's hear more about ants and long non-coding RNA.
And so thanks for the introduction. Let me just adjust things a little bit. First I would like to thank the organizers for giving me the opportunity to present the work here. So the project I'm going to talk about today is actually mostly done in the University of Lausanne. I'm currently working with the gene dosage lab in IMB Mainz to continue the investigation into this area. So I am an evolutionary biologist. I work on diverse mechanisms underlying sex determination across different animal taxa. And today the focal species is the ants. When we talk about sex determination, what comes to mind for most people is a pair of sex chromosomes. Especially it is very degenerated and highly iconic Y chromosomes carried by most males in most mammals. However, having a sex-limited chromosome is not the only way to achieve genetic sex determination. There are a few other options. And one of them is called haplodiploidy. Under this system, females are the diploid ones. They develop from fertilized eggs and they carry genetic materials from both the maternal and paternal side. On the other hand, males are haploid. They develop from unfertilized eggs and they inherit genetic materials only from the maternal side. Haplodiploidy have evolved multiple times across different arthropod taxa, and now it can be found in about 15% of all arthropods. That includes many important pollinators for human agriculture, such as bees and wasps, but also pests and disease vectors. So the idea of haplodiploidy is actually the first model of sex determination humans come up with. So back in 1845, Diazon, as shown in the picture here, a Polish beekeeper, he was the first one to notice that male eggs in honeybee do not require fertilization. But then we also know that ploidy alone does not account for everything about sex determination in honeybee, because occasionally diploid males can be collected. So based on a lot more observations, a complementary sex determination model was proposed. So under this model, all females are diploid. They carry two different alleles at the sex locus. They can reproduce without fertilization, and in that case, the offspring are haploid. Having only one allele at the sex locus, these haploid offsprings will develop into a haploid male. And while fertilization does take place, she will be able to produce diploid offsprings. If the diploid offspring inherits two of different alleles at the sex locus, they will again develop into a female. But when the diploid offspring inherits two of the same alleles from their parents, in this case, it will develop into a diploid male. These diploid males, they are sterile, they cannot reproduce, and they are a complete waste for the colony to produce them. So it is predicted that these will pose a strong selective pressure in the population to maintain multiple alleles at the sex locus in order to reduce the chance of producing these diploid males under random mating. So the gene underlying this complementary sex determination mechanism was first identified in the honeybee about 20 years ago. And indeed, it has many alleles. I'm showing here a tree containing 15 alleles, but in some other populations, the number of alleles at the sex locus can go up to much higher. The molecular mechanism for how this gene works was recently published. So this gene is a protein coding gene. In heterozygous individual, there will be two different protein variants and form a heterodimer. And in homozygous individual, which are the haploid individuals, and also homozygous individuals, there will be only one protein variant. And in that case, they will form a homodimer. And the configuration between the configuration of the heterodimer is quite different from that of the homodimer. Only in the heterodimer configuration where the functional domains of the CSD protein be revealed. And that would allow the turning on of female development pathway. So in order for this complementary sex determination to work in the honeybee, protein level interactions is crucial. Complementary sex determination is not restricted to the honeybee. From laboratory crosses, we know that complementary sex determination is the most prevalent and also the ancestral form of sex determination in Hymenoptera, which includes wasps, bees, ants, and sawflies. But the CSD gene in honeybee was actually a recent innovation restricted to the honeybee lineage, which means that for the vast majority of Hymenopteras, they rely on a different molecular mechanism for sex determination. And we are interested to uncover the molecular underpinning of sex determination in ants. So I guess we all have seen an ant, either in nature or in our kitchen. Ants are social insects which are known for their reproductive division of labor. If you open an ant colony, you might find different types of individuals. There are individuals such as this one with a large abdomen. These are the queens who are responsible for reproduction. There are also many other individuals in the colony who do not participate in reproduction, but in colony maintenance. Those are the sterile workers. But all these individuals that makes up an colony are females. And males do exist, but they are usually produced during a short period during the year. They are haploid. They do not participate into the daily maintenance of the colony. Males, adults do not even eat, so their main function is for reproduction. So for our project, what we did first was to identify a model species. And the species we picked was the Argentine ant, Linopisima humili. As its name might indicate, the native range of this species is in South America, but it has become highly invasive in many different parts of the world, including the south of France. So we got our laboratory colonies from the south of France. The main reason why we chose this species as a model for this project is because majority of the ant species do not reproduce in laboratory conditions, but this species does not seem to mind that. The production of new males and new queens can be induced in laboratory colonies by removing the existing queens. And mating within the nest between brothers and sisters happens readily, which is quite convenient for generating inbreeding crosses. And this is a relatively friendly species that do not bite or sting. So after we got our colonies from the field, we established 8 different inbreeding lines by doing brother and sister matings. And from all of the 8 inbreeding lines, we were able to collect sterile diploid males, which is telling us that indeed complementary sex discrimination is operating in this species. Afterwards, because this is a non-model species, there's not much genomic resources for it. So we started by generating a chromosome level assembly using a combination of nanopore long reads and Hi-C. So here on the Hi-C contact map, you can see 8 large scaffolds, which corresponds to the 8 chromosomes in the karyotype of the species. Then we did whole genome sequencing of the 38 diploid males we collected from the inbreeding lines, together with 142 of their sisters. With this, we were able to compare the heterozygosity differences between diploid males and females. So one of the hallmark of the sex locus under complementary sex determination is that females are 100% heterozygous at this locus, while males are 100% homozygous at the locus. With that in mind, we were able to identify just one locus in the whole genome where there's a striking difference in heterozygosity between males and the females. And when we zoom in, here is the heterozygosity of females in purple and males in green. We can see there's a small region of about 5 kb where females are 100% heterozygous, while males are 100% homozygous. So this is our candidate sex locus. Because we identify the sex locus within laboratory crosses, we also want to capture the sequence dynamic and diversity of this locus in the field. So we collected 86 haploid males from the field, which are the normal males. Then we sequenced the genome and estimated population genomic parameters across the genome. What we found was that the sex locus showed the highest level of nucleotide diversity across the entire genome. And with the same data, we were able to identify 7 different alleles at the sex locus. The size of the circle here corresponds to the number of individuals carrying this allele in the sequenced individuals. Then to understand whether all these 7 alleles are responsible for sex determination, we sequenced more females from the field, and we did some more inbreeding crosses. And here I'm showing the results. So any of the 21 heterozygous combinations among the 7 alleles resulted in a female development, while any of the 7 allele, when it's in a homozygous configuration, you always end up with a diploid male. And this demonstrates that the heterozygous combination among the 7 alleles instructs female development in this species. And of course, we're very excited to find out what genetic elements we can find within the locus. Thanks to the pioneer work in honeybee, we were expecting at first to find a protein coding gene. But to our surprise, there was no protein coding gene in the locus, which is represented by this black box here. The closest protein coding gene is still more than 10 kbs away from the locus. And what we have at the locus is a non-coding long-RNA. And the gene model of the non-coding long-RNA is here, with the exons of the non-coding long-RNA represented by these gray boxes here. The transcript of the non-coding long-RNA is about 700 base pair. The genomic region spans about 11 kb. So there are 2 exons of non-coding long-RNA located within the sex locus and the 2 exons outside. The transcription starting site is located also outside of the sex locus, which is indicated by our ataxic data with the open chromatin here. So because we identified 7 alleles at the sex locus, we were initially also wondering whether we were going to find 7 different transcripts of the non-coding long-RNA corresponding to each of the sex allele. But again, we were surprised by the finding. We found very little polymorphism between on the exons of the non-coding long-RNA. So here, each of the red bar represents a polymorphic site when you compare 2 representing alleles at the sex locus. There are many polymorphic sites in this region. However, all of the polymorphic sites are located outside of the exons of the non-coding long-RNA. This tells us that it is not the sequence polymorphism that would differ between males and females. And following that, we were wondering whether it could be expression level that differs between males and females. And to investigate that, we quantified the expression of the non-coding long-RNA over different developmental stage, from early embryo to first instar larvae and then to pupae. We compared individuals who are heterozygous at sex locus with those who are homozygous at sex locus. What we found is across all developmental stage, significant higher expression level of the non-coding long-RNA in heterozygous individuals when compared to the homozygous individual. Does that mean that our non-coding long-RNA, which is located in the sex locus, is at the top of the sex determination cascade? Before going on with more results, for the audience who are not familiar with insect sex determination, I would like to mention that there is a pattern. So different taxa would employ different instructive signals, which are different genes at the top of the sex determination cascade. But then all these different signals all go passed down through 2 very conserved genes. The first one's called transformer. The second one's called double sex. And the regulation of the instructive signal on transformer is always through alternative splicing. So there is a female-specific form and a male-specific form. Only the female form would be able to produce a functional transformer protein that leads to female development. So in our case, if the non-coding long-RNA is indeed associated with the instructive signal at the top of sex determination cascade, it should be regulating directly or indirectly the splicing of transformer, and the onset of its expression should start before the splicing of transformer is completed. And to investigate that, we compared the expression of our non-coding long-RNA and transformer over early development from fresh laid eggs to about 6 days post oviposition. What we found is that the non-coding long-RNA start to express about 48 hours post oviposition, which is similar to that of transformer. However, we also generated the gene model of the male-specific form and the female-specific form of the transcript of transformer. We can see here that there are 3 additional exons on the male form, which contains premature stop codon that eventually renders the protein non-functional. But these 3 additional exons also shows up as a higher band on the gel photo here. So we can see at 48 hours, when transformers start to be expressed, there's no splicing difference between females and the males. And around 72 hours, we can see a very clear alternative splicing of transformer. You have female form here and a male form here. And this happens after the start of the expression of our non-coding long-RNA. So from a temporal pattern point of view, it fits with the idea that our non-coding long-RNA acts upstream of the conserved sex regulator transformer. Then we would like to functionally validate the role of our non-coding long-RNA by performing RNAi. We injected double-stranded RNA against this non-coding long-RNA into very young eggs. And at the same time, we also did a control injection with this downcoating with the double-stranded RNA against GFP. Afterwards, we incubate the eggs for about 6 days. Then we extracted both DNA and RNA from the same egg. And with the DNA fraction, we could be able to tell whether the genotype of the sex locus, whether this egg is going to become a male or female. And with the RNA fraction, we check the splicing pattern of transformer. And here on this table, I'm only showing you the result from embryos that are heterozygous at the sex locus, which means they are destined to develop into a female. But about 10% of the individuals injected with the double-stranded RNA targeting our non-coding RNA, they showed exclusively male-specific form of transformer. Although the number of survived embryos were not high, this sex reversal of transformer splicing was never observed in our control injections. And the difference is significant, statistically significant, which gives us confidence that this non-coding RNA, which is again located in the sex locus, is involved in the regulation of transformer splicing. Hence the name ANTSR. If you've been wondering, it stands for ANT non-coding transformer splicing regulator. Just to summarize what we know so far about how this system works. So there are 7 alleles in the population. Each of the different allele is associated with this non-coding RNA ANTSR. And among different alleles of the sex locus, ANTSR only differ in intronic regions. In haploid individuals with only one allele at the sex locus, and diploid individuals who inherited 2 of the same alleles at the sex locus, there's a low amount of expression of ANTSR, and transformer is splicing to the male form and eventually leading to male development. On the other hand, if a diploid offspring inherits 2 of different alleles at the sex locus, that results in a much higher expression of ANTSR, and then transformer is splicing to the female form, and eventually you get female development. However, the exact mechanism of how this zygosity difference translating into regulating the expression level of the non-coding RNA ANTSR, and how this non-coding RNA regulates the splice pattern of transformer is something we are currently investigating. Lastly, we were curious about what's the origin of ANTSR evolutionarily. So non-coding RNA, the conservation of non-coding long RNA follows a different set of rules than that of protein coding genes. And they usually don't show high levels of sequence conservation. So the sequence can be highly divergent among closely related species. However, they do show conservation of synteny and function. A good example of that is the ROX1, ROX2 non-coding RNA from Drosophila, which are involved in sex chromosome dosage compensation. So to investigate how conserved is ANTSR, we looked at the genomic neighborhood that contains ANTSR, and across different ant species, but also in bees and wasps. What we saw here is a highly conserved gene identity and gene orders in this region. So flanked by the same genes, there's always a non-coding RNA. We also know that in 2 distantly related ant species, this locus is involved in sex determination. And also in a non-ant Hymenoptera, this locus is also involved in sex determination. So this could be repeated recruitment, but we think more parsimoniously, this locus containing the non-coding RNA ANTSR is likely the ancestral sex locus for not only ants, but more beyond in other Hymenoptera as well. And with that, I would like to thank my collaborators, Hugo, Claudia, and Laurent, and also the group members of my host labs. This work has recently been published, so if you're interested to know more details, you can read it over there. Also, yeah, I need to thank the institutes and funding bodies. And lastly, thank you for listening.
Okay, thanks a lot, Miya, for the wonderful talk. So let me remind you again, if you have any questions, you have to write it in the Q&A section, and then I can read it to Miya. As of now, there are no questions. I can maybe start off with one.
Yeah.
So I was wondering if ANTSR as other interactors or even other functions than messing up with transformer splicing? Is it
Not that we
known?
know of.
Or...
No,
Okay.
it's a new gene that we discovered during this study.
Okay. And also, I was also wondering how conserved transformer is in other species, because you checked on ANTSR, right? But what
Yeah.
about transformer?
Transformer is quite conserved across insects in its function and also in how it's being regulated. And especially in hemimetabolous insects, it's always regulated through alternative splicing.
Okay. Yeah, thanks. From my part, this is all. I don't think we have questions yet. Please write them in the Q&A. Okay, if there are no questions, then maybe we can stop here. And thank you, Miya, for the presentation. Thanks, Miona, as well.